Bacteria are the most primitive kind of living organisms found on the earth. They belong to the category of organisms which are prokaryotic and have been surviving on the earth since ages. They have seen the entire course of evolution as well. So, the very first kingdom classification which we come across is the kingdom Monera, which includes these bacterium. So, the general characteristics are they are first of all these bacteria it belongs to the kingdom Monera. So, you should learn the name as Monera kingdom. All the bacteria they belong to kingdom Monera. Now, these are primitive unicellular organisms, so they all are prokaryotic, smallest, and they can be found everywhere on Earth, from air, water, soil to living organisms, and even to the extreme climatic conditions. They can be found either alone or in form of colonies. For example, you can find just one bacterium all alone, or you can find a colony, a group of it found can also be found. They are the first primitive known organisms living on the earth. So, these are the general characteristics or just the brief idea about them. If we see the general characteristics of the bacterium, we find out or we see that they have a rigid cell wall. They can reproduce asexually by the process of fission. They lack the presence of nuclear membrane. This should be lack. Please correct it. It is lack of nuclear membrane and membrane bound organelles, a bacterial cell, and this makes them prokaryotic. A bacterial cell contains single chromosome which is not enclosed by nuclear membrane. Hence, this organism becomes a prokaryotic organism. And what is this structure known as? This structure is again another structure whose name is nucleoid. Some bacteria also have presence of chlorophyll and it is known as bacteriochlorophyll. This with the help of this bacteriochlorophyll they can form their own food, whereas the bacterium which lack the presence of bacteriochlorophyll they can have or they can perform heterotrophic mode of nutrition, that is they can form their own food. They can draw their own food by saprophytic mode or by parasitic mode. Saprophytic mode means they will be drawing their nutrition from the organic substances of dead and decaying parts of plants and animals, whereas parasitic mode it means it will be relying on a host and taking the nutrition from the host. The bacteria can be aerobic as well as anaerobic depending on the kind of environment they are present. They can, if they are present in an environment which is oxygen rich, they will be aerobic, whereas if the environment is lacking oxygen then they have anaerobic mode of respiration. If we see or classify the bacterium, we basically classify them on the basis of their different shapes. The very first category is of cocci. These bacteria are round or spherical. If you see them, they are basically round or spherical. They are smallest of all the bacterium known, and they can be present singly as micrococcus or in pairs. When two of the bacterium they are present together, we call them diplococcus. When they form a chain, it is known as streptococcus, and when a group is formed, it is staphylococcus. The different names or examples of diplococci bacterium is Streptococcus pneumoniae, Neisseria gonorrhea. These both are diplococci. We also have examples of streptococci, and they are like Streptococcus pyogenes, or staphylococci can be having grape-like structure, which forms clusters of cocci, and the example is Staphylococcus aureus. All these are scientific names, so if you are writing with your hand, you must underline them separately. I must do it properly. Streptococcus pneumoniae, Neisseria gonorrhea, Staphylococci, Staphylococcus aureus. So all the scientific names must be underlined separately. Next category is of bacilli. These are rod-shaped bacterium and they are quite small in size. Example is lactobacillus, which helps in formation of the curd. So these are the different shapes of the bacterium. You can just look at their shapes and you can understand that what is the difference. Cocci are all spherical or round shaped, whereas the rod shaped bacterium is the bacilli. You can see the structure. Spirillum is Vibrio, and spiral bacterium are different, and Vibrio is different. Don't get confused with these two structures. This Vibrio, I'm sorry, Vibrio, it's just written in spiral, but it is not the spiral bacterium. You can see it is comma shaped bacteria. Spirilla are the second one, which is Helicobacter pyrrole. So spirally are spirillum bacterium or corkscrew shaped bacterium. They are quite kind of S shaped, and they have presence of flagella, which helps in their movement. Helicobacter pyrrole is the example of spirally. Vibrio is a comma shaped bacterium, and a very common disease which is caused because of it is cholera. There are various shapes of bacteria present. They range from 2 micrometer to 0.5 micrometer thick. And the largest bacterium is around 16 to 45 micrometer, whereas the smallest bacterium is 0.15 to 0.31 micrometer. And you should know the denomination or how we write micrometer. Micrometer is written like this. Micrometer is written in this form, mu m. This is pronounced as mu. So I hope you will remember it. And when I have written here micrometer, it means that 1 micrometer is equivalent to 1 by 1000 of mu m. Okay. So this is just the little idea you should have about mu. The structure of bacterium is very simple structure. If you see the entire structure of a bacterium, you will see that the outer wall, first we will focus on these few structures. Outermost covering is the capsule or the slime layer. The importance of this slime layer is it helps in avoiding desiccation or drying up. So even if it is present in an environment which is completely dry, because of the presence of this slime layer, it can survive, or it can have the pathogenic nature alive within it. This is the importance of slime layer or the outer capsule. It is also protective in nature as because it is the outermost covering. Inner to it is presence of cell wall. You are aware cell wall is the outermost covering for the monerans as well as for the plants, and it is made up of peptidoglycan. So please remember that cell wall is made up of peptidoglycan. Next layer is plasma membrane, and it is a lipid layer. Inside it is the presence of cytoplasm, which contains the all the or which performs all the metabolic activities of the organism. The plasmid is the DNA of the bacterium, and it is capable of transmitting various factors from one bacterium to another. This plasmid is capable of transmitting fertility factor, resistance factor from one bacterium to another bacterium with the help of pili. Pili are finger-like projections which are present over the surface of the bacterium, and they are capable of forming cytoplasmic bridge when the plasmid is on the verge of getting transmitted, or yes, it is on the verge of getting transmitted. So pili helps in sexual reproduction of the bacterium. Chromosome or the nucleoid region is present in the center, dispersed. The nucleus is as such dispersed with the nucleic material or the nucleoid. It is called nucleoid because there is no nuclear membrane surrounding it, and it is just dispersed as such in the center. It is a primitive kind of nucleus which is present and is capable of performing all the metabolic activities of the or controlling all the metabolic activities of the bacterial cell. Now, not only that, the ribosomes are present in these structures, and these ribosomes are capable of protein synthesis. Food granules can be seen, and the food granules have the presence of digestive enzymes which digest the food which is inside it and take the nutrition from there. Prokaryotes also have a flagellum which helps in the movement. So, the movement from one place to another takes place with the help of these flagellum. So this is just structural description about the prokaryotic organisms. And you can see this various structures which I have already discussed. Peptidoglycan is present in the cell wall. Capsule is the outermost covering. Bacterial chromosome is the nucleoid contains only one bacterial chromosome. And plastids, some of the bacteria, they have presence of plastids, that is bacteriochlorophyll, uh, or chloroplast kind of structure, which is capable of autotrophic mode of nutrition and forming the food by its own. So the bacteria which have presence of this bacteriochlorophyll, they can synthesize their own food, whereas the bacteria which do not have the presence of this bacteriochlorophyll, they depend on heterotrophic mode for nutrition. Nutrition is of either autotrophic mode or heterotrophic mode. So autotrophic mode of nutrition takes place with the help of um, bacteriochlorophyll. These bacteria, they evolve sulfur. They evolve sulfur, and during the release of sulfur, what do they do is oxygen. In spite of oxygen, the release of sulfur takes place in these organisms, and they utilize the same solar radiation and the green color of the bacterium and release the sulfur uh, or anything else in response to the bacterium, or in spite of oxygen, they are releasing the sulfur. Chemioautotropic mode or chemiosynthetic mode is the one over where nitrifying bacteria they are found, and in anaerobic environment they are performing the function. Whereas heterotropic mode of nutrition is as such where they are depending both in phototropic as well as chemoautotropic or chemosynthetic mode, they are capable of making their own food. Whereas in heterotropic mode, they depend on someone else for the production of their food. Either they are saprophytic or parasitic or symbiotic. Saprophytic means they are depending on dead and decaying parts of plants and organisms for their food supplement, whereas the parasitic ones, they are depending on the host for withdrawing their food, whereas symbiotic ones, they live in harmony with the host and draw nutrition from there, and as a result provide them something else. So here something else is the nitrogen. Rhizobium bacterium is capable of fixing atmospheric nitrogen into ammonium, and the ammonia is taken up, or ammonium ions, they are taken up by the plants, because nitrogen requirement is there for the plants. Whereas rhizobium bacterium, what it needs is the food, and food is given by the same plant over which it is surviving, or the host plant. Next is about the movement. As the bacterium, you have seen that they have presence of this flagellum. With the help of flagellum, they can swim, and whip-like movement, like you can think of a fan moving left and right, and that kind of movement is seen by the flagellum for movement in water. Whereas some of the bacteria, they can just fly or they can float in the air as such with the help of those dust particles. Respiration is required for the process of getting food, or by which they draw their nutrition. So aerobic mode is seen in the bacterium which depend on oxygen for the production of the food, whereas anaerobic bacterium, they can form their food even in absence of oxygen. So depending on the presence or absence of oxygen for the nutritional requirement, the bacteria can be classified as aerobic bacteria or anaerobic bacteria. The mode of reproduction is basically binary fission, in which one cell is there and it divides. First of all, the nucleus divides, forms two nucleus. Then, after the cytoplasmic constriction is formed, and the invagination of the cytoplasmic constriction results in formation of two daughter cells. This takes place during the favorable condition. What about the unfavorable condition? During that time, we see the formation of cyst, and there are two ways by which a cyst can be formed. You all can see that a sporulation is being explained over here. You can see a cell wall is there. When this is the site where I am trying to, yes, this spore coat. At first, whenever the unfavorable condition arises, So during that time, the first thing which happens is this pre-spore is formed, which has a septum and a mother cell is there. Along with it, there is presence of this entire nucleus within this area, and the nucleus, it starts dividing. If I'm telling the inside story, it's that the nucleus is dividing continuously, and as the nucleus divides, little amount of cytoplasm is held along with it, and this entire cell has several outer walls present. Because of the presence of this kind of encysment, it is very difficult for the unfavorable condition to harm the inside spores, and as a result, when the favorable condition arises, this outer wall is broken off, and the germination of the or growth of the spore into the bacterium takes place in the favorable condition. Similar situation is seen in case of encystment. You can see a mature cyst, and how the nuclei is getting divided, several cytoplasmic divisions are formed, and finally when the favorable conditions will arise, it will burst off and release all the trophozoites. So although these are just manipulations in order to make you understand, basically this is just in order to survive the unfavorable situation or climatic condition. Now the so this is all about the bacterium and a general idea that how does the bacterium survive.